Okay, well maybe I should just start then. Welcome to the talk. I am very grateful to the organizers and to all of you for coming. I believe that future historians, when studying the moment in time when humans seized control of their own evolution as a species, they will highlight this meeting. This is like the Asilomar meeting for molecular biology. Next slide. Okay, so in this talk, I've geared this talk for non-experts. So one goal is that for most of the people who don't work in this research area, you'll come out of this with a sense of where the state of the art in the field is. If you are a researcher in the field, none of that will be new to you. You might be slightly bored. But I do have a few things which are at the frontier that I'm going to gesture at, and those are the things that if you are a researcher in this field, I would love to chat more about with you. Okay, so it's going to be a little bit of a mix, so just be prepared. Okay, so let me just start with these things, which again, if you're not an expert, these things might be a little bit new to you, but I'm just throwing them out there so the experts, when they're bored looking at graphs that they've seen before, they can ponder these things. Okay, so this is a new scientific subject, and despite the fact that we are commercializing the application of polygenic scores, there are a lot of really fundamental questions about the genetic architecture of complex traits in humans that still are unanswered, and they're actually deep scientific questions. They're not shallow scientific questions. So one of the surprising things is that as far as anybody can tell right now, the genetic effects that are making one individual human different from another individual human are approximately additive. So many, many papers are written by teams, including us, trying to find nonlinear effects, trying to find locus-locus interactions in the genome and failing to do so. In fact, a very good paper just came out from the Inouye group at Oxford on the use of neural nets to try to find these nonlinearities, and they failed to do so. So there's something quite interesting about the architecture, which is quite surprising to most molecular biologists and wet lab biologists, that again, we're not saying that genes don't interact. We're not saying that genes don't interact. We're saying that specifically when we look at individual differences within our species, those differences seem to be driven by a fairly simple genetic architecture, which is additive, which is linear. And that's a deep mystery for why that's true. There are hypotheses for why that's true, but it's still kind of very interesting. Related to that is a point I want to make, that we really do not know. There are very few cases where we actually know what the causal variation is in the genome. We can localize it to the point where we have tags or signals that allow the machine learning predictor to make accurate predictions, but that's not the same thing as knowing actually at a molecular level what's happening. And we really need, I think, better knowledge about causal variance before we attempt editing. Okay. Now, Gene Smith has he has a slightly different view, which is like, oh, well, if you're not sure, maybe just edit all of them or edit some of them, and you can calculate some expected gain because you never know in each particular edit case you actually did edit the causal. But so you could still make some estimate for where you can get to. But I do think, like, as a topic of investigation, causal variance is something that a lot of energy needs to be focused on. Something we've worked on in my lab is limits to genomic prediction, scaling laws with data. It looks to us that at least with the set of algorithms that people are currently using, everything in our field is data limited. So it's basically just if you give us more data, we can see that the predictors get better, the models get better. And with the exception of height, there isn't any other complex trait which is close to saturation. So height, we've sort of reached the limit of our ability to predict, at least with common variants. But for every other trait, whether it's breast cancer risk, prostate cancer risk, diabetes, et cetera, we can show you plots that estimate how much better the predictor will get if the number of cases that we have in the training set, goes up by a factor of 2 or a factor of 10. So still, we have not reached the limit of our ability to predict. Probably all of you know that progress in Gen AI has been driven by these things called transformers. It's an open question whether, I think you're going to hear a very interesting talk about it at this meeting, whether that new way of trying to do genomic prediction will yield some gains that the older algorithms did not give us. So that's quite interesting. Some Complex traits that I think are super interesting and definitely doable in the sense that there's enough heritability there and potentially enough data to do these things, but it just really hasn't been gathered yet sufficiently. And of course, everybody here is familiar with cognitive ability. I think there's pretty good evidence that things like big five personality traits, when you correct for the noisiness of the measurement of the phenotype, that corrected heritability is quite high. It could be as high as the heritability of this trait. Okay, so eventually we will be able to predict things like personality traits from DNA. And one that's super highly heritable, just think of identical twins having almost the same face. This one is totally doable if somebody gave us if say all the people in 23andMe took photographs of themselves and we pushed those photographs through face recognition algorithms, which then map the face into about a 100-dimensional space. Each of those parameters in the 100-dimensional space that characterize your facial morphology is itself super heritable. And so we could probably predict face from DNA quite well if we had the data, but we just don't have the data. Next slide. Okay, so where are we as a company? We've genotyped a little over 120,000 embryos, so it's just not an insubstantial number. We learned a lot about the IVF industry from this, attitudes toward embryo screening, where things are heading. So I just want to say a few things for the other companies in this space or in adjacent spaces. The hardest thing is earning the trust of these IVF professionals, the clinics themselves. That's the gating function. You cannot win in this space by impressing venture capitalists, impressing rationalists. You need to actually make progress in signing up clinics, having them trust you, having their doctors recommend your product to the general public passing through their clinic. Okay, I think people who are in this space should publish their research. Okay, I do not want to end up in a situation where company X is claiming they have a great predictor for Y, but they can't release the secret of why their predictor is so awesome. And it sort of degenerates into a low trust situation where people are making claims that aren't testable. It would be much better if we evolved into a high trust situation where people could be confident that the companies in this space have published their research, have demonstrated their technical capability in a transparent, legible way, and any claim that they make on their website or in a venture deck is something they're willing to stand behind with a hostile statistical geneticists questioning them. Okay, so that's where I would like the field to evolve to. Next slide. Okay, so here's a kind of picture of the world of the repro IVF world. So in the IVF clinics, you have people who really don't know anything about genomics. They might know a little bit about genetics, but they don't really know anything about very large genomic data sets, machine learning on large genetic data sets. They even don't really understand validity testing. So in other words, you train the predictor in population A, and then you have an out-of-sample population. You test on that population. Even that kind of stuff is challenging for a typical IVF physician or a genetic counselor at the clinic to really deeply understand. So you've got this population. Then you have a population of researchers. This is actually a large field now. So about 10 papers a week, more than 10 papers a week, are published with the word or on the topic of polygenic prediction of human traits. And the number of authors, if I just took a guess, would be anywhere between 5 and 50 for those papers. Okay, so if you add that up and you think these are all people with academic positions, typically, some at companies, that's a very big pool of researchers, right? And very few of those researchers are actually here at this event. Why? Because they're sort of they're not allowed to think about reproduction, right? They're only allowed to think about preventing disease, understanding the genetic architecture of diseases. They don't think that much about reproduction, but they are able to test your claims. So if you say, hey, I can predict X with AUC of Y, this whole population is going to look in at what you're doing and say, like, these guys are lying because I have a bigger data set. We have a specially funded data set that we built to do that, and we can't predict that well. What are these guys talking about? Okay, so people in this world should be aware that eventually the crows are going to come home to roost. If you make crazy claims, eventually people are going to challenge your claims. And it's, in the short run you can win, but in the long run I do not think you're going to win that way. Okay, so publishing on X, publishing in a PR video doesn't count. Next slide. Okay, this is the AGI thing, which I think Tsvi already mentioned. So we're up to probably 1% of GDP now being spent to create the AGI. So 1% of US GDP is being plowed into creating the AGI. Could be dangerous. We might be in a full-on race situation with China to do it, throwing safety out the window. Maybe smarter humans have a better chance at solving the alignment problem or making these new machines safe. Or maybe smarter humans have a better chance of surviving if they're not safe. For all those reasons, if like when you leave this conference, you talk to your housemate and you're talking about your P-doom, you might say like, well, can you guys just like take 1% of the amount that you're spending to make the unsafe super machine? Take 1% and try to improve humans, right? So that would still be billions of dollars a year spent on what we're trying to do, whereas right now it's a much smaller sum. So anyway, but I think everybody here gets this point. Okay, next slide. Okay, so let me just review this field very quickly, and I'm just going to show you what I think are some of the best slides or graphs which illustrate what has been done in this field and sort of where we are. So just to level set, your genome, 3 billion base pairs, whole genome sequence is an attempt to read out each of those base pairs. Less expensive technology, a gene array, sort of like what 23andMe uses. You're measuring about 1 million loci in the genome. So it looks like you're getting much less data, but the arrays are specifically designed to measure the loci that are likely to vary between individuals. If you look at most of our DNA, my DNA and his DNA, we only vary at about 1 in 1,000 positions. So there's only a few million base pair differences between him and me. And that variation is concentrated at known locations. So the arrays are deliberately trying to read out the information in the most efficient way possible. Okay? So when someone says, oh my God, they only use arrays, we use whole genome sequencing, there might be advantages to using the whole genome sequence, or you might just be paying for a lot of additional information that is not interpretable, or actually there isn't particularly that much information because the extra letters you read out are the same in almost everybody. Okay, so just a distinction to be made. Next slide. Okay, so typically what are we doing in this field? We're doing machine learning, or AI is a fancier term. Typically, analysis is something like, maybe we have 100,000 cases, so those would be like 50 to 70-year-old people who have diabetes. Those are cases. And then we might have several times that number of controls. And the machine learning is basically trying to figure out which regions of the genome are predictive of the case status. Okay, so that's a simple disease trait. Now, you can use whatever method you want. You either succeed or you don't succeed. But what's important in this field is the rigor of the validation of the predictors. Okay, and number one, and this is something that like critics who are not informed about our field often mistakenly claim that the evidence that these predictors work is weak, but in fact, I would argue it's actually stronger than almost any other kind of study that's done in biomedicine, because the statistics are way higher. The validation is done out of sample. So quite often you're validating on a population that grew up on a different continent than your training population. Quite often you're validating on a population that is actually a completely different generation. Right? So you might have trained on people who were born right after World War II, and you're validating on people who were born in the 1970s. Okay? And yet the predictors perform about as well. The validation result doesn't seem to change if I change the geographical location on the planet, or I change the generation, when they were born, et cetera. So it seems pretty robust. And the ultimate kind of validation that we do is using siblings. So we actually, there's enough data around that you can assemble. In our case, we have this sort of standard validation cohort that we use, which is about 20,000 sibling pairs. Okay, so we're comparing individual sibling 1 against sibling 2. They were born and raised in the same family. Okay, so there's a lot of commonality in the environments that they experienced. And the question is, are you able to predict which of these two sibs is more likely to have diabetes, or which of these two sibs is taller. And so that's a pretty rigorous form of validation, right? So I think you're getting pretty confident that you really are predicting genetically driven individual differences between those sibs. And those sibs are exactly analogous to the embryo problem. Because when you're doing embryos, you have a bunch of sibs. And you're trying to decide which of these embryos, which of these sibs is going to be taller. Which of these sibs is going to be higher risk for prostate cancer, right? So in terms of the rigor of the validation, I would argue it's very, very high in this field. Okay, next slide. This is the growth in the number of papers written. So you see in recent years, the number of papers which are on the topic of polygenic risk scores is in excess of 10 a week. Big collaborations, usually involving multiple universities, publishing in this area. You know, it's a mature and very vibrant academic discipline. Okay, next slide. Next slide. Next slide. Okay, so here's roughly what you can do with these predictors. And, you know, this article was actually about us, and it's quite old now. It's like this was written, you know, 5-6 years ago. What can you do? You have a group of people, and you're only given their DNA, okay? And you say, I want to classify people as high risk, ordinary risk, or low risk for a particular disease condition. Left here, these are all men, coronary artery disease. On the right, only women, breast cancer. And you can see, and these are cohorts where we have the whole medical history of people over this age range. And you can see a woman who's said by us, because of DNA, to be high risk for breast cancer, has a materially far, far, far higher chance of having breast cancer than someone that we characterize as low risk for breast cancer. So this is a very material difference, right, in your life. And so is this, okay? So if people say like, oh, these polygenic scores, there's no proof that this is actually going to help you select a better, you know, a healthier child. Well, I just would say I don't understand the thinking that leads to that process if people see data like this. Okay, next slide. Okay, this is a height prediction. This is our original paper on this from like 2017, 2018. We can predict height to within a few centimeters of standard error. What's interesting here is the genetic architecture. So if you connect your genome, the 3 billion base pairs end to end, you lay all the chromosomes end to end. So you have, and then you put a coordinate on here like 0 to 3 billion. These little lines, if they point up, I think it means the minor allele at that site makes you slightly taller. If it points down, the minor allele at that site makes you shorter. And so this is the full-blown genetic architecture of human height. And there are about 10,000 sites in your loci, regions in your genome, that the predictor needs to know the state of in order to make the prediction. Okay? And so this is one of the most complex human traits. IQ, or cognitive ability, is probably a bit more complicated than this in terms of the number of loci involved. They're spread out all over your genome. You know, when we published this paper, the people who had been in this field before we came along were totally blown away because they just didn't think this was possible. But automated machine learning prediction can produce this. Okay, next slide. This is a validation of our paper from another group at Stanford, and similar thing, predicted height, actual height. Okay, next slide. Okay, next slide. Okay, so this is an example of predicting probability, the risk of actually developing the condition. And interestingly, once you have the predictor, you can build some very simple theoretical models, which is the red line. And they can ask, like, if I divide my cohort into bins where the people in each bin have a particular PGS score, risk score, how well does the empirical data, the blue dots, conform to the theoretical prediction? And I would say pretty damn well, okay? This suggests we have some mastery over like what is going on here, right? Now, if you're low risk, you can see the absolute probability that you end up with, in this case, hypothyroidism, close to 0. If you're a high risk outlier, you could have, 0.7, 0.8 probability. So when you're born, I will tell you, you're going to have hypothyroidism, right? That's what this is saying. If you're far enough an outlier on the risk score, just from your DNA, I can say to you, yeah, you know what, hypothyroidism is in your future, okay? So I would have to say this is actually like science fiction, right? I mean, to most doctors, if you show this to a doctor, they'll be like, wait, this is not real, right? This is just, you're making this up. No, this is actually, we do this every day with embryos, right? The only reason this is noisier is just we just didn't have as much data from which to draw the blue dots from. If we redid this with a larger set of women for whom we knew the breast cancer history and had their DNA, we could basically fill this out exactly the way that this fills out. Okay, next slide. Next slide. Next slide. Okay, I mentioned already that we use sibling pairs for the gold standard validation, and roughly speaking, we can say that the predictors perform just slightly less well on siblings than they do in random pairs drawn from the population. And it's because there is some sort of confounding, like we're removing some environmental stuff that mistakenly the algorithm picked up as genetic signal. But when you do it with siblings, that goes away and you lose that slight bit of predictive power. But generally things are basically almost as predictive for SIBs as in just with random pairs from the population. Okay, next slide. Next slide. Next slide. Okay, now one crazy criticism that we hear, and you can open up the New York Times or Bloomberg or like high prestige journals writing about embryo selection, and they'll quote some quote expert, usually a doctor or a bioethicist, making this like crazily stupid statement that I don't see how there can be much value in computing embryo risk scores because after all, they're all siblings. They can't differ very much from each other, right? So now if you think about your high school, your friends growing up, like surely you had a friend who was like really tall and his brother was like below average in height, right? There's some, even though families are similar to each other, there's still quite a lot of variation between the siblings. And in fact, like anybody who is like reasonably high IQ and had a good high school biology course can immediately figure out that the variance goes down by 2 because you've pinned half the genetic variants are the same. And if you have additive variants and you've pinned half of them so they're the same between the two SIBs, you've reduced the total variance by a factor of 2. The standard deviation goes down by square root of 2. So what we plotted here is pairwise differences between, you can see we have a lot of data. The blue dots are pairs of random pairs drawn from the population. The red dots are pairs of siblings. And so you can see the siblings cluster together more than random people chosen from the population, but there's still huge amounts of variation between siblings. And so there's this crazy like non-quantitative word cell logic, like wait, how different can they be? They're brothers, right? So what's the point of like comparing the DNA of one to the other? But what you're doing is you're still working with this amount of variation and you're reducing it when you select the embryo. Okay, next slide. Okay, slightly theoretical point. One thing that was surprising to molecular biologists, people who learned from James Watson and Francis Crick and all this other stuff, these guys had a very strong prior that only genes matter. Like, oh, how many genes? There are only like 20,000 genes, and like if it doesn't code for a protein, what does it possibly do? When you build these predictors, you notice huge amounts of the variance accounted for in these predictors is outside of genic regions. It's outside of regions that code for proteins. So there's a whole bunch of stuff going on that, like there are various communities that are still very resistant to this message, that genes and protein coding regions of DNA are far from the whole story, but you can really see it explicitly if you actually try to build predictors. Because you see that if you, for example, confine yourself only to input information, which is information about the genetic letters in the genic regions, you would lose like half the predictive power. Okay, so that was a little surprising at a kind of theoretical level for a lot of biologists. The other prior that people have, which turns out to be totally wrong, is about something called pleiotropy. So What is true about pleiotropy is if you look at a particular important gene and you mess with it, okay, say I mess with this gene, it will affect a lot of different things, okay? So that is a form of pleiotropy, like one gene affects many phenotypes. That is a true statement. A false statement is to say that I cannot alter trait 1 without simultaneously altering trait 2, 3, 4, 5, 6, 7, 8, 9, 10. And this is a very important question for genetic engineers. Anybody here who's a budding genetic engineer? In the worst case scenario of overwhelming pleiotropy, you would be in trouble, right? You want to go in and make an edit and you want to do something to the baby, but it does like 10 other things at the same time. The amazing thing about the genetic architecture that we're discovering is that most traits that we look at are roughly disjoint in the support of where their variance is located in the genome from the others. So I literally can modify your diabetes risk without changing your height, without changing your IQ, without changing your extroversion, okay? And if you do a little calculation based on the data that we have, you can see there are roughly 1,000 different phenotypes that can vary more or less independently that characterize an individual human. It's about 1,000-dimensional space that you can explore. So gene editors are set to explore that 1,000-dimensional space, and indeed you can navigate your ship in each of those 1,000 dimensions roughly independently if you want to. Okay? Not sure if what I'm saying makes sense to you, but the point is this pleiotropy thing could have been an obstacle to controlled genetic editing, control of evolution, but when you look at the data, it's actually not. There's plenty of independent variants for each independent trait that you care about that you can edit at will without changing the other traits. Okay, still very poorly appreciated by people outside this narrow area. Okay, next slide. Next slide. Next slide. Next slide. Embryos, next slide. Okay, so in the global IVF industry, there's about, I think this number is a little bit low, but it's about 2 million embryo genetic tests per year worldwide are performed. The most common test is something called PGT-A. It's an PGT aneuploidy, so preimplantation genetic testing for aneuploidy. Okay, that's the most common test. And what you're looking for there is an extra chromosome or some kind of gross rearrangement of your chromosomes, which is very problematic. And in the US, about 70% of all embryos undergo PGT-A screening. So almost every clinic in the US will recommend PGT-A screening. The main reason they recommend it is that if you transfer without knowing it an aneuploid embryo, that embryo is not likely to lead to a successful pregnancy. Okay, so you want to like just remove those from consideration and only consider the euploid. Euploid is the opposite of aneuploid. So you want to only consider the euploid embryos for transfer. And so that's why this particular screen has become the main one that people do. We designed our wet lab process to attach to this. So people are already doing this. We take the same biopsy, and we're able to, because we amplify the DNA and then we run it using an array, we're able to both do the aneuploidy characterization, but then using that same data, we can compute all the polygenic scores as well for the embryo. So we're sort of trying to attach to the existing procedure. And so our cost is not that high. It's roughly, I think, $2,000 to $3,000 per cycle, depending on how many embryos you have if you're doing both A and P. We don't control the pricing. The clinics control the pricing. So we can control like what they pay us, but we can't control what they charge you. And they would love to charge you as much as they can. Okay. You can think of like any IVF clinic or like minor surgery outpatient clinic as a little business. You know, if we're having an abstract conversation with them, they're like, yes, we understand your PGT-A screen is much more accurate than the one we're currently using, and yes, we understand that would lead to a higher percentage of successful cycles, blah, blah, blah. But can you cut your price because these other guys are charging less? And like, no, we can't cut our price. It's like, well, we're going with these other guys. So think about that. Think about Hippocratic, what was that thing? Yeah. Well, think about that bullshit. So these guys are saying like, oh, I know your thing is better. I know your thing is better. I know the family saved up a lot of money to come in here and get a baby. And I know your thing, I agree with you. Okay, we're having this conversation with them at ASR, American Society of Reproductive Medicine, or at their clinic. They're like, we agree with you. Your test is better for PGTA. It will lead to a better outcome on average in terms of having a baby for our clients, but we're not going to offer yours. And why? Because we want to make more money. So we encounter this every day. You're never going to be allowed to release this video, by the way. So, but this is the real world. Okay, this is the real world. Okay, next slide. So we're quite proud of the fact that our PGTA, because remember we're measuring 1 million different loci on the genome. So if something funny happens with a chromosome, that part of the chromosome, we have many individual data points in that region. So if you sort of rearrange some chunk of chromosome or you put an extra copy in, we can see it like, you know, it's very easy for us to see it because we have so much resolution in the data that we gather. So our PGTA is way more accurate than the old method for PGTA. And so there was an independent study done by this guy who is at a clinic called POMA, which is in Seattle, near Seattle, Kirkland, Washington. And he presented his results at the ASR meeting, I guess 2021. And I'm sorry you can't read that very well, but if you use so he did random assignment of embryos to using our PGTA screen and the two major competitors. And you can see the miscarriage rate is way lower if you use our screen, and the pregnancy per IVF cycle is way higher if you use our screen. And again, so to knock the doctors again, like they don't care. Like they're like, oh, I believe this. I know Klaus. You know, I went to school with Klaus. He's awesome. Yeah, these results are probably right. Yeah, we're not going to offer your screen. Okay. So she's taking a photo. So no, you can't. You can. I'm kidding. I'm just kidding.
Published result, right?
Yeah. This is published. Yeah. Klaus has published this result. So now what's going on here is that these methods are noisier, and so they occasionally will classify a good embryo as bad, and they will occasionally classify a bad embryo as good. And in both cases, it hurts the family because they're either like an embryo that they could have transferred, they think they can't transfer because the test gave a false positive, or an embryo that they think is good actually is bad, and when they transfer it, it's not going to result in a good pregnancy. Yeah.
How close is the price point to where you think they would be willing to pull the trigger there?
We're very price competitive with the old these NGS technologies. So it's not really necessarily, so sometimes our competitor will really cut the price to a certain level, but sometimes it's not even stuff like that. Like it could be like weird kickback things happening, or it could be like some of our competitors also supply like the fluids and materials, media that these people use in their labs and stuff. So they're very complicated business relationships here, but clearly the patients the doctors, the clinics don't have as their like #1 goal optimization of this parameter. They don't. Okay, next slide. Okay, so you guys know all this. Let me go to the next slide. Okay, so these are the different tests that are offered by IVF clinics. So PGTAs, this is the best example, like you just have a trisomy of an extra chromosome, but also you can get what's called the structural rearrangement. PGT-M is for Mendelian. So if the mother and father are carriers, they know that they are carriers of some mutation, which could lead to a bad genetic disease. What we can do is we can track the state of whether this mutation gets passed down to the embryo. Okay, so that's pretty rare though, because the only people who are doing that are ones who already know that they're carriers. And then the one that is of most interest to this meeting is PGTP, in which we sort of know something about the genetic architecture of a trait, and we measure enough stuff that we can predict for the embryo where it is on that trait. Okay, and so the frontier is PGTP. We're the only lab that has a process that does all of this at once. So we can do all of this with the data that we gather from the embryo. Okay, next. Okay, this is a little bit more about this question of, because one of our competitors who might be here, I don't know, did you really invite those people? Oh, they might be here. Okay, so what is the advantage of whole genome sequencing, right? So, you know, I'm in a waiting room, and you know, I have a meeting with Elon at 2 p.m. And someone's in there talking to him, and she walks out. What's her name again? Noor. Yeah, Noor walks out, and then I go in, and he's like, I'm making this all up, okay? He's like, oh, but I heard they do the whole genome, and you're only using an array. So isn't her product better than your product, right? So again, what I said earlier, the array is specifically designed to extract the most useful information when it comes to computing the polygenic score. You are getting more information if you do a whole genome sequence, but it's not clear what difference that actually makes for computing the polygenic score, okay? And because the data used to train the polygenic predictors is mostly array level data, okay? So a lot of this is marketing. Now, if Kian is here, he'll talk a little bit about our partnership that was recently announced between his company. His company does whole genome. Oh, there he is. Yeah. So awesome. So they do whole genome sequencing. Originally, when they started their business, it was whole genome sequencing of adults. Some of those adults want to have kids. If so, they refer them to us. And then instead of us taking saliva from the parents to get array genotypes of the parents, we'll already have the whole genome sequence from nucleus. And so then we'll handle those patients with, you know, that initial data set already there. And because there are phenotypes that we don't compute, we don't predict any cosmetic traits, we don't predict cognitive traits, nucleus does, we will pass the data back to nucleus to make those additional, compute those additional scores that we don't compute. Now you might ask, why do we not compute those scores? Because the media, you know, for a long time was claiming we did, but we actually don't. For us, in building a business, I've sort of emphasized that everything is about the clinic. You have to make the clinic happy. They have to trust you. This is a new weird technology, et cetera. And if we went more aggressively toward polygenic scores like intelligence, there would be a lot of clinics that just wouldn't want to work with us. So for us, the trade-off was these health benefits are substantial. We want to, and the aneuploidy, better aneuploidy prediction is substantial. The benefits are substantial. We don't want to jeopardize offering that to as many clinics as possible just to offer these edgy things. So, and we've been around longer, so that was our decision, and we've stuck to that decision. But we can always partner with other entities to do that additional computation of those other traits. So that's kind of what our position is. Now, I just want to say something about de novo mutations in embryos, because ORCID claims, so for example, if you see this, there's a journalist called Ashley Vance, and they just made like a 20-minute documentary, very nicely done documentary about Noor and ORCID and family, you know, tech titan family in San Francisco who just went through ORCID. It's a very nicely done documentary, but you'll see Noor claiming that one of the big benefits of what ORCID can do is detect potential de novo mutations in the embryo. So that would mean the mother and father are not carriers of this particular genetic mutation. It occurred de novo as a mutation, copying error, what have you, in the embryo, and they claim they can detect it. And I challenge them to justify this claim, okay, for the following reason. They're amplifying, just like we're amplifying, from only maybe 5 cells worth of DNA. Think about that, the DNA from 5 cells. We're amplifying it. At the most optimistic, I would say the whole genome sequence you derive from that data has an error rate per SNP, per base pair, of maybe 1 in 1,000. So at least 1 in 1,000, maybe more like 1 in 100 times, you are calling the wrong letter at that locus. Doesn't matter for polygenic scores, because for a polygenic score, you have thousands of input variables, and if like 1% of them are wrong, you have an error in the polygenic score, which is probably like 1%. if you're calling a new de novo mutation, which are very rare, there's only a handful of de novo mutations per generation, okay? You're trying to find something very rare, but you have a base error rate of 1 in 100 or 1 in 1,000. Almost everything you call is a false positive. She says they can do it. I'd like to see a conversation between their bioinformaticist and a patient and say, well, yeah, you know what, this letter could be dangerous. I want to warn you about embryo number 4 because this letter could be dangerous. There are probably like 1,000 dangerous letters in each whole genome sequence because of the base error rate, and they don't know where those errors are. Okay, next. Okay, so here's just a very simple example. Maybe Alice, in her family, there's a history of breast cancer, and she's very concerned about it. She goes through IVF. After the aneuploidy screen, she has 5 euploid embryos. Certain people would say, well, there can't be any difference in breast cancer risk between these siblings because after all, they're all related to each other, right? Next slide. Okay, so we go ahead and compute the breast cancer risk for each of the embryos. In this example, one of them is high risk, one of them is low risk. Okay, and there's some spread. And we've actually published papers on this using embryo data showing like what the spread is. But you could actually have predicted what it is theoretically. Next slide. Okay, so if their main concern was breaking that chain of high risk in the family for breast cancer, they might want to prioritize this embryo for transfer. But we're not excluding this embryo. I mean, so some people criticize us for causing embryos to be destroyed or discarded. We're not telling them they can't use this embryo. We're just making them aware that our best estimate of the breast cancer risk status of this embryo is far higher than this embryo. That's essentially the information that the family are getting. Okay, next slide. Okay, so yeah, and yeah, next slide. So, and then you can do studies where you see what the risk reduction is as a function of how many embryos you had, the quality of the predictor, and picking the number 1 ranked embryo versus the say randomly selected one or average one, how much risk reduction you get. And so we've published a bunch of papers on this. Okay, next slide. Let me just go through this. This is just like what the report looks like, just in case you're curious. You can just slowly. These are like the 23 chromosomes. This is the overall health score. The health score is computed by summing up the absolute risks for each of the diseases in the index. And the coefficient, which is not computed by us, it's just taken from the public health literature. It's the life expectancy impact, the negative life expectancy impact of each disease condition. So it's a reasonable way to weight the risk that's inherent from that prediction. The coefficient is the life expectancy impact. And we just compute that all up and we get something called the embryo health score. We publish papers about how the embryo health score is predictive of longevity. So better embryo health score, you have a longer longevity prediction, and lower is worse. And so, the data comes out like this. And these are in standard deviation units. Okay, next slide. Turns out if you just select on embryo health score, you actually get risk reductions across all the different conditions. Next slide, next slide. This is what the report looks like. Can't really read it. There are 11 embryos on here. I think these are 11 euploid, 2 aneuploid. The embryo health score is here where the physicians demanded it be because they didn't want to discuss all the individual risks with the patient because that takes too much time. They demanded that we just have one score because it's fast. You can just go in the room and say, this is the one we recommend you transfer. Yes?
Do you regularly have conflicts between certain disease propensities in one embryo versus another, and it's like kind of a toss-up?
Yes, very good question. So part of the reason why the embryo health score was developed is in the earlier version of our report, where we just gave bang, 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 these are your risks for all these different conditions. You're an outlier, positive outlier for heart disease, but you're a negative outlier for diabetes or something, right? And then, of course, the parents would be asking the genetic counselor or the doctor, which one is worse? But I'm only 97 percentile for this. And the doctors didn't know what to say, and the genetic counselors didn't know what to say. So they're just like, can you just give us one number? So that's why we were forced. We were literally, I mean, it was a good thing because it was good research for us to do and we're happy with it. But we were literally forced by them to develop this embryo health score for practical reasons. Now, our report contains everything. So the geek parent, nerdy parent can go 50 pages in and look at like everything. But the physician just wants to have like a top line thing. It's like, we're transferring embryo 4. Thanks, good luck, you know. Lovely to see you again, Mrs. Smith. Okay, next. Okay, so that's the report, the report, the report, the report. Just keep going. Okay, here's some new, oh yeah, go ahead.
Yeah, just, you mentioned that the scoring is kind of all positive, right? Or like it's all kind of going in the same direction.
Not sure what you mean by that, but.
Like lower score or higher score is better. That you don't have like big cross-cutting.
Well, the EHS can be negative. So if your embryo has like high propensity for lots of diseases, it will have a negative EHS score.
Right.
Yeah.
But I guess what I'm asking is was it surprising that you didn't end up in a situation where it's like two different risk axes are kind of
Anti-correlate, yeah. So we do not find, and again, this is like very core scientific knowledge about genetic architecture of human health, which again, like, I don't know, there's some deficit of deep thinkers in this field because like people don't really remark on this, but on our team, we find this very deep information, right? So it could have been the case that there are two well-known diseases where if you're high risk on one, you're likely to be low risk on the other and vice versa. It could have been the case that we discovered that. Just as like in the definition of G, general intelligence, it could have been the case that high spatial ability was negatively correlated with high verbal ability, but it doesn't turn out to be the case. So there is actually effectively a G factor for health in the same way that there is a G factor for the manifold of cognitive abilities. That is new news. Yeah. Like the people in epidemiology, public health did not know this. We published it, you know, but you can go look at the paper. I mean, anybody who can read can look at the paper and go, well, I didn't know that was going to turn out to be true. Wow, it's neat to know that that's true, but okay.
It seems surprising.
It ain't theoretical physics. Like they're not like necessarily getting what you tell them. So, okay, but that is true, and it is a deep thing. I have no explanation for you other than like, you know, a plausible hypothesis. Like there are some important systems in your body that all affect things, and that is why there's some positive manifold of correlations. Okay, that's my hypothesis. That part's not substantiated by like deeper evidence, but that's my hypothesis. Yeah.
There are a couple of different biobanks that are out right now. Like the most famous one is DK Biobank, but there's also like FinGen and a couple others. Have you noticed any pros and cons of using different biobanks for assessing polygenic risk scores?
Oh, I could go to okay. So we use data from all of them. Okay. That's why also like I think a criticism, like you might say, like, oh, somebody earlier in one of the sessions said, oh, they're just looking up things from whatchamacallit catalog, PGS catalog, right? You want to, if you were hiring a consultant, you know, to help you interpret this stuff and do some additional computations, you would want somebody who has access to as many of these data sources as possible, is used to doing things like, oh, let's compute a predictor in FinGen and then we'll validate it in UK Biobank. Let's compute a predictor in all of us and then we'll validate. So our lab is doing stuff like that every day, right? But not everybody is doing that, and not everybody has access to all of these data sets. So of course, yes, you want to see, because there are weird idiosyncrasies in this subject. And so if you don't know them, you could make some mistakes.
Yeah, like FinGen in particular is like codes.
Fins are a little bit weird, right? Okay. So you basically queued up this point, which is that until recently, the best predictors for any particular disease condition or phenotype were always trained on European ancestry populations, and they work best in European ancestry populations. And if you took one of those predictors, just because there's more data, there's more training data, if you took one of those predictors and you tried to use it on an African American person or a Japanese person, there would be a significant decline in predictive power. And it's mainly due to something called linkage disequilibrium, or the correlations between states of SNPs. The patterns of correlations between state of nearby SNPs are different in the different populations. So what is a perfectly good tag SNP for the causal SNP in the European population might not be a good tag SNP for the true causal SNP in the Japanese population. Okay, so we think that's the reason for the degradation. We think, but we don't know, that the actual architecture of causal variants is the same, approximately the same in all the different populations. But even that is a hypothesis, so that we don't know for sure that that's true. Okay, but for whatever reason, there's a non-transportability property of current predictors outside the original training population, ancestry group. Okay, I mean, going from England to France is fine. Going, white people from England to France is fine, or France to, Texas is fine. But Japanese, Chinese, Nigerians, a problem. Okay.
In American context, with black people, is very difficult because GDK populations simply aren't that many.
Well, and yeah. So it's all data. It's all data limited. Now, we collaborated with the Taiwan Biobank, and we got access to. Now they're up to about 1 million. When we did the analysis, we had like 500,000. So we published for the first time the only predictors that work as well in a non-European ancestry group as the best European ancestry predictors. Chase has a question.
Are those new predictors rolled out yet?
Have they been released to the general public?
Yeah, like if,
No.
if I like.
They have not been. But
When will they be?
good question. So this paper, we submitted, I think, what is the final revision to Nature. So once it appears, then there will be a whole database made available of a whole bunch of results from the training. Who asked that question? Oh, anyway, yeah. So yes, soon, hopefully. But we're waiting for the publication of the paper, and then the stuff will come out. Okay. But this is big news because, again, like until just recently, you could not do polygenic prediction in non-white groups very well, and now you at least in this group you can do it. Okay, next slide. Here's a related paper, same collaboration. This one is just predicting height in Taiwan, and you can see we can predict height pretty well. Okay, next slide. Okay, next slide. Oh, next slide. Okay, I'm just going to talk about some fun stuff. How much time do I have left? Okay. Everybody know what this is?
Yeah.
Everybody love this picture? No. Isn't this one of the most beautiful pictures in science, which, like, once you see it, you have a deep understanding of something that, like, 90% of the population cannot possibly admit is true. Okay, so Project Talent, I think it was like, this was back when America was functional. We were fighting the cold. What's that?
5% of all high schoolers in 1960.
Yes. Okay. So this was when America was functional. We were fighting a formidable opponent in the Cold War. They had launched a satellite into space before us called Sputnik. And we reacted with a whole of society effort to compete, as opposed to now where we just cope and pretend China's not actually kicking our ass. So back then they did things like, oh my God, let's test 5% of all ninth graders in the United States of America. And let's give them a very nice instrument which measures their math ability, one which measures their spatial. What is spatial ability? That's like it's even like crime think to talk about something like what's spatial ability. That can't possibly be something called spatial ability. And then the other one is verbal. Okay. Now what happens when you graph all these kids? You get this ellipsoid. This ellipsoid has a dominant major axis. If you needed to compress someone's general intelligence into one number, you might as well take the coordinate along this major axis because sort of does a good job. Not everybody with the same coordinate on this major axis has exactly the same scores, obviously, but like this direction is kind of better and this direction is kind of worse. And of course, what I was saying earlier about health is that health seems to actually have some kind of property like this. Yeah?
When you say like actual IQ, are you like adjusting for?
Oh, don't read this. Don't read. Yeah, see, you're an expert. Don't read this because these slides were not made for experts, so this is really like kind of like EA, which really isn't IQ. Don't read this part yet. I'll talk about. I just wanted to show you the picture. I wish I had taken that text off. This is an old slide, but okay. Next slide. Okay, here's a great paper. Belsky et al. Proceedings of the National Academy of Sciences, 2018. There are 4 longitudinal cohorts. A longitudinal cohort in social science is a group of people that may have been followed since they were like in, I don't know, junior high school until they're 70 years old. Okay, and each of these longitudinal cohorts is several thousand people. Okay, so sociologists identify a bunch of kids, sign them up, they're following them, they have their juvenile delinquency records, their grades, what they got locked up in prison for or not, whether they admit to using dope, on their survey. They have all this information about every person in this cohort. And like as we approach the modern era, they genotyped every person in the cohort. Okay? So you know everything about the lives of these people. These people are Wisconsin longitudinal study. Dunedin, who knows where Dunedin is? Any Kiwis here? Okay, so we have Kiwis, Wisconsinites, Harvard, I think this is Harvard retirement study. Health in retirement study. Yes, this guy knows everything. And then we have Ad Health. So these are Americans. These are specifically Wisconsinites. These are specifically Kiwis.
Very different generations too.
Yes, very different generations. These people were born, some of the cohorts, the people were born 20, 30 years before some of the people in the other cohorts, right? So if we have any sociologists, social scientists here, all your criticisms are taken care of, okay? All of them are taken care of, okay? Now, does the IQ or EA predictor predict what happens to these kids? Okay, that's the question, right? Okay, so now we have to, what do all these numbers mean? Okay? On each of these panels, if I go to the right, that's a higher polygenic score. If I go to the left, it's a lower polygenic score. Vertical axis is this is an outcome score of their lives, the individual person. And this was created not by evil geneticists. This outcome score was created by the sociologists before the genetic technology even existed. So they just said like, oh, -5 if you went to prison, -3 if you're a drug addict, +1 if you graduated college. They just have some way of scoring. Like, did you have like a classically kind of good life, or were you like, you didn't have such a good life, right? So totally, reasonable. If you looked in at the scoring system, you would say this is a reasonable scoring system. It was not made by the evil geneticists, okay? So the social scientists did this, the geneticists did this. Notice what is happening here, okay? So the polygenic score seems to predict the outcome that the social scientists care about. Now, the panels are born in a low SES family, socioeconomic status, born in a low SES family, born in an average SES family, born in a high SES family. So even within the rich population, having a high polygenic score was good. Among the poor people, poorly educated parents, having a high polygenic score was good. That's what this graph says. But just let me say one more thing before I ask questions. This blue thing, that blue diamond, is the average polygenic score of the people in the panel. So interestingly, high SES people have on average higher polygenic scores. People from low SES groups have on average lower polygenic scores. It is the social Darwinist nightmare involving genes, validated with, kind of massive statistics, good enough for social science. It's been replicated. James Lee replicated it again. Yeah, so of course we live in a society where we don't really want to learn and update on stuff, but it's kind of proven beyond a shadow of a doubt, what is really going on here. Okay, now I'll take questions. Yes?
How common is it for an Asian to
have a shitty PGS score in white biobanks, but a good PGS score in Asian biobanks?
I would actually not, what's the right way to say this? I would just prefer not to compute the Asian PGS score using a predictor trained in a white ancestry training population, right? So, of course, you can get all kinds of crazy stuff because of the different LD structures, right? So, I would just prefer not to even like address that. Maybe we'll talk about it offline, but I don't think it's very informative. The answer to your question.
And also, do they often implicate the same gene, but just different locals within the same gene?
Yeah. So all the evidence across ancestry group GWAS is that the regions that are implicated are the same. So most people would say my zeroth order hypothesis is it's the same causal variance for every large ancestry group that are driving individual differences. The question is just whether a particular tag SNP that your machine learning algorithm picked up for that causal is still a good tag SNP in the other distant population. And most people in this field believe that is the cause of the decay in predictive power when you transport the predictor. Does that help? Okay. Again, hypothesis, like this is ongoing science. This is why, like, if some guy comes in and is like, this is all solved, man, by my gene editing kit. It's not all solved. There's like real science that needs to be done here. Yes?
Yeah, I'm just curious. So just eyeballing it, it looks like, especially in group A there, the low socioeconomic status people, the outcomes seem less correlated between the polygenic scores and the outcomes. Is there a reason for that? Like the low socioeconomic status?
Yeah, I mean, there could be, these are different societies, different eras, even like 70s kids, 50s kids. So, yeah, you mean like here? Yeah, I mean, it could be like, oh, this slope is not the same as this slope. There could be like statistically significant differences across these different longitudinal groups. I've not looked into that in detail. I'm just asking about gross features of whether, like, oh, is some of this genetically influenced? Yes, okay.
Bad question. I'm just curious.
No, it's a good question. I don't really know the answer, but it's not surprising that stuff like that could happen. You, sir.
I don't know if there's enough information to tease this out, but is this increase a cause of absolute increases in education project score or relative increases?
I'm not sure what you mean by that, but
like, let's say now you have a society of like everyone between IQ of like 50 to 100, however you find IQ.
Okay. 50 to 100, that's not good.
Okay. And then you have another society that has like IQ between like 100 and 150. Would you still see like these same effects? Like,
You're asking if it's a linear correlation?
I'm asking whether it's like a function of ranking in society or a function of actual like added intelligence.
I don't know the answer. That's beyond the research of these guys.
Sort of
But
know the answer for some things. Like for educational attainment, the polygenic score, for example, in Estonia and in Hungary, it became much more predictive after the Soviet Union fell. Like the context does matter for these things.
yeah. If that's all you're asking, yes, context does matter in these things. The lady who is building an amazing artificial womb, and if you're at this meeting, you should ask her about it.
I'm curious about the polygenic score. The averages.
Yes.
It honestly makes me sad. This is true. This data. But so I want to understand, like, if it's positive by 0.5, what does that mean? Is this a standard deviation?
Standard deviation. Yeah.
And then I was also wondering about relative versus absolute risks. So when you say someone's at the high end of risk for X disease,
Yeah. So
what are we talking about?
when we compute the embryo health score, we are actually trying to estimate from the polygenic score the absolute risk. Absolute risk. So lifetime risk that they'll develop this condition. And that is then multiplied by the negative effect on longevity or quality life years. And then we sum it up.
So, okay, so an example, let's say like 25% of the population will get some kind of cardiovascular disease.
Yes.
And you say this embryo has an 80% high risk of cardiovascular
Yes.
disease. Am I going to multiply that 25 times 80%?
Right. So there are two embryos, right? One embryo is like at the population average. So he's going to have 25% chance of. And the other one is an outlier for heart disease, and he has 80% chance. And then let's say the negative effect is minus 10 years of longevity. So the contribution to the sum for embryo 1 is minus 10 times 0.25. And for the other embryo, it's minus 10 times 0.8. So it's actually linear in the absolute risk. Sir.
I was going to say two things. training sample, except for you get like a sharp drop with Africans. There's just like a big split there. The second thing is, I wanted to ask about the coding variant thing you said a lot earlier in the presentation.
Yes.
Because when I do like Mendelian randomization analyses with Tsvi QTLs, I often find results that are very different from when I do them with coding variants. And I would like to know why you think that is, if it's not actually a problem.
I'm not sure I have a good answer to your question. You know, I'm not saying that the way in which non-coding variants enter into the predictors is the same as coding variants. I'm just saying that, and I can operationalize this pretty concretely. For a while, Illumina was selling exome sequencing as a product. Okay, why? Because it's much cheaper. You only sequence the coding region. It's like a few percent. So it's way cheaper than a whole genome sequence. But then they would say to the investigator, the naive investigator, well, you know, everything's happening in these coding regions. This information, like in the exomes, you don't need, right? And the exome is all you need. And but then, like when you actually look at what you need to compute a polygenic score, you need far more than the exome data. And that's the only point I'm trying to make. The details of like really what is the nature of the causality for variants that are in the coding region and outside is very complicated. I don't know the answer.
And I will say, like, they're far more likely to be causal variants, at least in my common fine mapping tools, if they are in coding regions than if not.
That I believe. Right. Okay.
If you think you have this portability problem, and you think that the causal variants are probably shared, not too different between populations, does that mean the new Chinese data sets or new data sets from other distinct populations are particularly useful for identifying causal variants?
Yes. So if you ask like, hey, Steve, in your lab, what's the #1 thing you guys are working on right now? It's trying to identify causal variants because we have early access to a well-powered population study in a distant ancestry from Europeans. And we're using that to try to like zero in on where the causal variants are.
And does that become useful only if you start doing some sort of editing, or is it useful?
No, okay, yes, great question. So the first way it will become useful is you might be able to build a fully portable polygenic risk score. Where like I don't care about your tagging, blah, blah, blah. I have the causal variants, and this will work as well in each of these populations. Yes.
This is effectively what you get with African samples. Sub-Saharan African samples have much more limited LD structure, and if you get a polygenic score based on them, it tends to generalize incredibly well.
Yeah. So anyway, this is all the like, if you're in the weeds in the subject, this is the stuff that we work on.
Excuse me if this is an uninformed question, but I was really curious when you talked about the the predictability really requiring you to look at the non-coding regions of DNA. How does that work? Like, what is the mechanism? Like,
Yes, good question, good question. So as a kid, you were probably taught about this, some of the biologists know, what's this thing called? The thing that Crick and Watson, fundamental synthesis, or what do you guys call it? Central dogma, yeah. So when you're a little kid, they teach you the central dogma, right? Which is like proteins, DNA, RNA, proteins, right? And so, it must only be the coding regions that matter because that's what determines the protein structure. And all of this stuff in the middle is potentially junk DNA, okay? That was a story for a long time. And you can still like go to Berkeley and tap on the door of a biologist and they will repeat that for you, even though it's 100% contradicted by the data here. Now, if you have some intuition for information theory, okay, you would think to yourself, wait a minute, wait, this mouse has the same number of proteins as I, and even the worm has almost the same number. There must be other variables, other switches that are at play. And so there are probably little switches, DNA switches, which are not themselves modifying the protein, but they're modifying the expression of the protein, the rate of the extent to which that protein is expressed. They are regulating the interaction between different mechanisms. So you can imagine a lot of other handles on the development of the organism which don't actually require you to modify a protein. And that's why I believe the stuff in the middle is not all junk DNA, and it's the stuff that shows up in our predictors.
It obviously has to be, because that's what like splicing the QTLs and methylation QTLs are.
Yeah. Now much is known about this, but when in my childhood education I was taught this junk, and I just, if you just think about it, it can't be true, actually. So, question?
Yeah, I wanted to get into more of like the economics of this, right? Like you mentioned, it's about like 2 or 3 thousand per IVF cycle, like
Yeah.
presumably on top of the normal IVF
Correct.
costs, right?
Correct.
So, in terms of like whether, like how does that sort of cost compare to the expected benefit of doing this selection in terms of like healthcare costs or income or?
Oh, wow, wow, okay. So there were slides I skipped where these were actually presented to the NHS, this little calculation. You know, if you genotype your whole population and then you compute a bunch of scores, you can show that the system actually will save a lot of money. Like the genotyping of your population will pay for itself very easily in sort of public health applications. Is that what you were asking about?
I mean, like for, let's say for like a parents who are considering this, right? Like, is it worth it?
Oh, for parents.
them? Yes.
Okay, well, there are various ways to think about this. One way to think about it is like, oh, if I have 10 embryos and I pick the best EHS score, I'm getting about 4 extra life years, or at least a few extra life years for my kid. And what's the value of an extra healthy life year of your kid?
Yeah.
So a few $1,000 bucks.
When you say you have 10 embryos, like not all of those will successfully implant, right?
Yeah.
So
Right, so I mean, like, look, someone like Gwern can build you a little calculator. It'll have a slider, maybe 2 sliders. You can decide whether it's worth it to you. We're not trying to like, it's up to you whether you think this is worthwhile. Let me just show 2 more slides, just for fun. Okay, similar study to the last one we showed. This is tracking kids through their grade 9 through 12 education and also even into their later careers. And you characterize them by their polygenic score, which is color-coded. Blue is a high polygenic score for EA. It's a cognitive kind of thing. Yellow is a low score. So you're tracking these people through. Conditional on having the yellow color. Very likely you stopped at this level of math. Very unlikely you went to college. Conditional on being blue. Pretty good chance you took calculus in high school. Pretty good chance you went beyond college in your education. Again, only DNA information. Only information that you could possibly access for an embryo before deciding that it would become your child. So, what's that? Yes, everything in this field replicates because it's not bullshit. Okay, okay, next slide. Okay, now, maybe you've seen this picture before. These are the chickens that people eat. These are the common breeds used in 1957, the breed from 1978, and a breed from 2005. They are photographed at the same age. These chickens are the same age, and they've all been given as much to eat as they want and the same food. Okay, so again, all the low IQ, social science criticisms of this shit, it's all been taken care of, okay? They did the smart thing, okay? Now, in the population of these chickens, there's not a single one that looks like this, okay? Like maybe you have 1 million of these guys, you might find one of these guys, but all of these guys look like this. Yeah, question?
Well, quick question. So you're controlling, for the food, are you controlling the type of food they're eating?
I think they're all eating the same food and they can eat as much as they want. Okay. These are agricultural scientists. They're not tricky guys. They're doing the reasonable thing. Okay. So how did this happen? What happened? Were there Martian genetic engineers at work in this period? No, it was just selective breeding of chickens to give you plenty of Arnold Schwarzenegger breast meat. Okay, so that's all that happened. But now if you are a genomicist and you work in animal or plant models, they build predictive models too, just like we built, okay? They predict things like milk production, how much lean beef, how much fat is in the beef, how big are the breasts, how fast does a chicken mature, how many eggs does it. They track all that stuff, and they can predict that stuff even more accurately than we can predict our stuff because they have very inbred populations. Okay? But it works. It works, and you can make a super chicken if you want to. The method is up to you. Okay, but you can definitely make a super chicken, and you can look in and see, like, why is this the super chicken? Where in the genome does it differ from the not so super chicken? And you can figure all that stuff out. And so this is a mature science, and people should pursue it. Thank you very much.